Okay, so now we are going to talk about lymphocyte activation, more specifically about B cell and T cell activation. So with respect to activation of both B and T cells, even though they are two different cell types and even though they do two different things, they have a similar pattern of activation. So we have first, of course, our antigen. And so everything starts with the recognition of an antigen. So here's our virus with our proteins on the surface, our little spike lollipop proteins. And so the first step in activation of our lymphocytes is binding. And binding takes place at the B cell receptor, which the B cell receptor, of course, can bind to the virus proteins by themselves or the virus antigens by itself, and the T cell receptor, which can only bind to antigens that are in the form of peptides bound to MHC molecules, as we've discussed. We also have co-receptors that will be involved, and we have everything occurring with binding happens at the surface. And so the cell on the outside will have these receptors, of course. And then the second step is going to be in actually being able to transduce a signal to tell the T cell or B cell you've bound to your antigen. So signal transduction, and it is very similar to all other signaling pathways in many other systems in your body. It basically occurs intracellularly, of course, and with respect to our immune system, this is going to require what we call ITAM motifs. And an ITAM motif is simply just an immunoreceptor tyrosine-based motif. And you see tyrosine, and tyrosine is used by many signaling pathways. Insulin receptor is a tyrosine kinase, for example. So it's a very commonly used pathway. So it's an immune receptor tyrosine-based motif. Now these ITAMs are also going to be dependent on tyrosine kinases. And as we know, kinases are going to phosphorylate. And so this is going to be a signal that comes from an antigen receptor. So sometimes phosphorylation is a way to control the function of the proteins, the enzymes. And in this case, we're activating the enzymes. So phosphorylation of these enzymes are going to be, or these proteins is going to be activating them. And then we have an amplification stage. And this is where you're amplifying the signal. So we have some downstream secondary signaling molecules. Some common ones are protein kinase C. We have calcineurin. And we have MAP kinase pathway. So you're going to see these come up as we go through this talk. And then all of this is going to end on transcription factors. And the transcription factors for our lymphocytes are nuclear factor, or NF, kappa B. B is for beta. We also have NF, that's right, NFAT, nuclear factor again. And this is a nuclear factor of activated T cells. So nuclear factor kappa beta, nuclear factor activated T cells. And we also have a third transcription factor, which is AP1, transcription factor activator protein 1. Now these three are all going to be in B cells and T cells, and they are going to turn on different genes. In a B cell, it's going to turn on genes that are important for antibodies. And in a T cell, of course, it'll turn on genes that are important for T cell function. And then finally, you have your cell response. And keep in mind, of course, that even though there are similarities between how B and T cells get activated, their actual response is very, very different. Okay, so let's look a little bit more carefully at the molecules that are involved in B cell and T cell receptor and signaling. So I have here a B cell receptor, and I have here a T cell receptor. Now, a B cell receptor, as we've mentioned, can see antigen directly. So it can bind to these little blue circles directly. A T cell receptor cannot see antigen directly. It has to see it associated with an MHC on another cell, and I'm not drawing the other cell right now. And so that blue circle will look very different to a T cell. It'll look like a peptide. So it's a peptide antigen. So keep in mind, the T cell receptor cannot bind to the round little circles directly. They have to bind to a modified version of those round little circles. And the B cells can respond to those round little circles. And these round little circles are referred to as external stimuli. Now, after you get binding of your B cell receptors cannot signal. So keep in mind, BCR, TCR cannot signal. And so this is kind of a form of control. If they could signal that any peptide or any antigen they bound to, you would automatically get that cell activated. There are other requirements in order to get these cells activated. And one of those control mechanisms is to have the signaling molecules different than the actual binding molecules. So B cell receptor, T cell receptor are binders, and then we have our signaling molecules. So on the side of our B cell, we have Ig alpha and Ig beta, and these are our signaling molecules for the B cell receptor. And on our T cell side, we have our zeta chains, and we have our, we're going to have a gamma epsilon chain, and we're going to have a delta epsilon chain. D. And these are the CD3 complex. Zeta chains are also considered part of the CD3 complex. Now, what's important about this is the T cell receptor, again, cannot signal, and so it's the CD3 complex that will signal for the T cell receptor. The other thing to point out are these green ITAM motifs. So remember, the ITAMs are simply the immunoreceptor tyrosine-based motif. So this is going to allow these receptors to bind to and interact with tyrosine kinase enzymes, which is going to be important because it's the tyrosine kinase enzymes that are really going to get the signal transduction pathway going. All right, so let's talk about B cell activation. So B cell activation requires a few things. We have our B cell receptor that is binding to this little round blue ball, as we showed in the previous slide. But we also have this funny little purple arm. So let's talk just for a moment about what this funny little purple arm is. We have the signaling apparatus here. So this is the B cell receptor signaling. Oops. And I'm just going to put this here right now. If you have the B cell receptor binding to this blue ball, it's going to signal through Ig alpha and Ig beta, its signaling apparatus, and this is going to be signal 1. Okay, but this is not enough to get your B cell activated. Your B cell needs these co-receptors. Oops, they're purple. So these are the co-receptors. And this equals signal 2. So what is the purpose of signal 1 and signal 2? Well, one way to think about it is signal 1 So if you think about B cell activation, one, it requires signal 1, so it must recognize the antigen. And then signal 2, there must be some signal to identify that this is an actual external antigen, that there is an infection. So you can think of it as sort of an inflammatory signal. And it's basically because most of our antigens, when you're healthy, are self-antigens, and you don't want to activate B cells to self-antigens. So typically when you're healthy, you don't have these inflammation signals floating around because there's no inflammation. When you have an infection, your innate response is going to release those inflammatory signals. And so the innate immune response is going to help the B cell understand that I see this antigen and I know we're infected, so I need to be activated. So for B cells, this particular signal comes from our complement system. So complement are a series of proteins, so they are proenzymes. They are in your blood, and they get activated. And so the one that is important for B cells is complement component C3D. Okay, so that's all you have to remember here. So this little red thing here is complement component C3D. And what's important about C3D is you can see that it's attached to the blue ball. So C3D is able to attach to antigens, and it does this in a way that it just requires certain charges on the antigen surface that make it look different than our cells. There's certain things that make the antigens look different than us, and the complement system takes advantage of that. And so C3D will bind to this blue ball. Now, C3D doesn't care if the blue ball came from a virus or it came from a fungus or it came from a bacteria. It can just bind to the blue ball. And by binding to that blue ball, the complement will trigger a response in this co-receptor because CD21 is complement receptor 2. Okay, so complement receptor 2 is on the surface of your B cells. It binds to the complement component C3D, and that sends signal 2. So this is signal 2. Now after your B cell has signal 1, signal 2, then it really starts to get going. Now you're going to want to bring in some tyrosine kinases to get everything going. So the first one that's going to come in is a SRC. It's SRC. So here's our tyrosine kinases. And number 1 is SRC. And SRC is going to find those ITAMs. Okay, so SRC binds ITAMs. And SRC is a tyrosine kinase, so it's going to phosphorylate those ITAMs. Now, once those Ig alpha, Ig betas are phosphorylated at their ITAM motif, a second tyrosine kinase, which is SYK, is going to bind Ig alpha and Ig beta. So we'll draw SYK, we'll use a darker green or lighter green. So here will be SYK, and I'll just write it for Ig beta, but it'll do the same thing on Ig alpha. Now SYK is also a kinase, tyrosine kinase, but SYK is really what's going to activate the downstream molecules. And it takes several steps to do this, but we're just going to draw the big steps. And so once you get SYK recruited to this apparatus, you are now going to get 3 major downstream pathways. And you're going to first get you're going to get a RAS pathway. Now, RAS is a G protein, so it reacts with GTP. RAS is going to activate the MAP kinase, similar to what happens in insulin signaling. And then this MAP kinase is going to activate the transcription factor AP1. And so that is activated protein 1. Now, SYK is also going to take advantage of some molecules or some fats you have in your membrane. And we have phosphatidylinositol in our membrane, PIP2. And so via phospholipase C activation, PIP2, PIP2 will become IP3, inositol phosphate 3. And this is going to activate calcium. And then the calcium is going to activate calcineurin. So this is our second pathway here. Oops. And then you're going to get nuclear factor of activated T cells, transcription factor. And our third pathway has to do with a byproduct of clipping off inositol phosphate 3 from phosphate inositol, and that is DAG, diacylglycerol. And DAG will activate protein kinase C. And so this is our third pathway. And then this will activate NF-kappa B. And so now we have all of our 3 transcription factors. So we have our NF-kappa B, which came from diacylglycerol and protein kinase C. We have our NFAT, which came from inositol 3-phosphate through calcineurin. And we have our activator protein 1, which came from RAS and the MAP kinase pathway. Okay, so let's now look at what's happening with our T cell receptors. So our T cell receptors are a little different than our B cell receptors, of course. Our T cell receptors require peptide with MHC on it. This is a CD4 response, so we have a CD4 molecule, which is in this purple. And so CD4 will kind of glue together the MHC molecule and the T cell, as long as the T cell recognizes the blue peptide. We also have here, just in block forms, our CD3 complex. We have the gamma epsilon and the delta epsilon chains, and we also have our zeta chains. And so CD4 is really going to have the motif, which will recruit the first tyrosine kinase, and that tyrosine kinase is LYC. So LCK will come along and interact or be recruited by the CD4 complex. And LCK is going to phosphorylate the ITAMs. So you're going to add phosphates to these ITAMs, and you're going to add them to your zeta chains as well. So those are going to get phosphorylated by LYC. Now, after LYC has phosphorylated those ITAMs, we're going to get our second kinase, which is ZAP70. And ZAP70 stands for zeta chain associated protein kinase. And so ZAP70 is going to come in and interact with our CD3 complexes. And similar to our SYK in our B cell story, ZAP70 is what's going to initiate those 3 downstream signaling pathways. So I'm just going to put here ZAP70. It's already been activated. And we're going to have those same 3 pathways. We're going to have our first pathway, which is going to give us RAS. and we're going to get the MAP kinase. Let me make this a little smaller. And then we are going to get our AP1. And then ZAP70, similar to our SYK, is going to clip phosphatidylinositol using our phospholipase C, PLC. And we're going to get IP3, which is going to release calcium from our intracellular calcium stores, and through calcineurin, we are going to get NFAT. And then PIP becomes DAG, diacylglycerol, and that's going to activate protein kinase C, and this is going to get our NF-kappa B. And so again, we have our 3 transcription factors, and those are going to go to the nucleus and turn on T cell specific genes. Now that we understand the signal transduction pathway, we can start looking at drugs, and we can start understanding how they work and what their targets are. So one of the oldest drugs used for transplants is cyclosporin. And cyclosporin's target is the nuclear factor of activated T cells, or that NFAT. Tacrolimus is also for transplants, and tacrolimus also targets the NFAT. The mechanism of action is, of course, to inhibit calcineurin activation. And the result is going to be no interleukin-2, which is downstream of your NFAT. So NFAT will turn on the interleukin-2 gene. And this is going to lead to a decrease of proliferation and a decrease in growth of your immune cells. So your immune system can't divide and respond to the transplanted tissue, for example. And so this is going to be used primarily for tissue organ rejection. So both of those drugs kind of work the same way. So I'm going to draw a line there. Now we have sirolimus, which is also known as rapamycin, and this is going to block IL-2. And it does this through targeting another pathway. It targets mTOR. mTOR is sort of a master regulator for autophagy, for self-eating, meaning that if your cells are under a period of stress and they don't have enough nutrients, they will actually start to eat themselves. And so by interacting with this mTOR, you can force the immune cells to eat themselves if they get activated, and so they don't destroy the tissue. And the result is sort of the same: no growth, no proliferation. And this is also going to be targeting organ rejections, but it is used for kidney transplants. Okay, and we also have a basal Lixi molecular antibody. So when you see this MAB, that means it's an antibody, it's a molecular antibody. So they make this in a lab and then they inject it into people. Now what this is targeting is you're targeting the IL-2 receptor. So it's an antibody, targets IL-2 receptor. And then it's going to bind and inhibit the receptor. And so IL-2 is a major growth factor for activated T cells. And so a lot of these drugs for transplants are going to have some sort of pathway to inhibit T cell activation or slow it down or do something to suppress it. And so this molecular antibody will block the IL-2 receptor. The IL-2 receptor, since we're on it, has 3 parts. It has 1, a gamma chain, 2, a beta chain, and 3, an alpha chain. And the alpha is referred to as CD25, and this is the high affinity. So T cells will have on the surface oftentimes the gamma and beta, but that doesn't really do much. It really is the alpha that brings all 3 of them together to form the high affinity IL-2 receptor. And so the result of blocking the receptor is the same: no growth, no proliferation. And so this one is going to also be for kidney transplants. And again, it targets the CD25. Sorry, I want to make sure you can read that. And so it's, again, kidney. Now, this is not used alone. It's going to be used along with a combination of drugs, but it's been approved for use for preventing kidney transplant rejections. Azathioprine is going to inhibit purine synthesis. And of course, if you're a rapidly dividing cell, such as an immune cell responding to a foreign antigen, like a foreign tissue, such as a kidney, you're going to want to divide. And in order to divide, you have to have purines and pyrimidines. And so if you block the synthesis of purines, you're going to block the potential for cells to divide because they can't make enough DNA to do so. And so this prevents replication. Oops. And so this is going to be kidney also, but also it can be used for rheumatoid arthritis. And then we also have mycophenolate motile, and this is a specific inhibitor of guanine synthesis. Now this has the same effect as your azathioprine. It prevents replication, but it has less side effects. And this is also going to be used for kidney transplant. So this is kind of the same. This is kind of the same. Okay. And so you can see that our immune suppressive drugs are targeting a lot of the pathways that have to do with T cell, either activation or replication or proliferation and expansion and growth. Now, last I want to just talk about control of T cell activation. So when we think about control of T cell activation, we have to think of it as on 2 levels. One is to tolerize, so to inhibit self-reacting T cells or B cells. And the other is to control an activated lymphocyte clone. Okay, so let's talk about the first one because we should be able to understand the first one now. So let's talk about the first one. The first one is through tolerance through energy. And energy just simply means non-reactive. So the first part that happens here is you have a T cell, and it interacts with an APC, let's say. And so you have on the surface a T cell receptor, and you have on the surface an MHC molecule, and you have some peptide that the T cell is specific for. Now this T cell is seeing the peptide. So T cell receptor plus peptide, MHC, equals signal 1, right? But there is no signal 2. Just like our B cells had to have that signal 2 through complement, T cells in a synapse also have to have signal 2 through your APC, and that would be through a molecule called CD80. So I'm going to draw CD80. This can also be CD86. And then on the T cell receptor side, it will always express CD28. But now, similar to our B cell story, our MHC does not sense that there's a foreign pathogen. There's no inflammation. There's no inflammatory signals. So it does not express on its surface CD80. Instead, the T cell, which sees the antigen and is looking for that CD80, doesn't find it. And so what happens here is you get a T cell that's anergic. And basically what happens is instead of the T cell receptor, now it upregulates FAS. And so it will eventually get killed by a process called apoptosis. And so this is a way of tolerance. And so this is a peripheral tolerance. We also have central tolerance, but that doesn't happen, of course, in our bodies or in all of our tissues. Peripheral tolerance can happen in every tissue. Central tolerance only happens in the bone marrow for B cells or in the thymus for T cells. Okay, so let's talk about the second set of control, which is after activation. We really don't need to guess this. Well, this is kind of B, and then this is kind of C. Okay, so when we talk about activation, we're going to talk about a kinetics. Kinetics has to do with time. So we'll have time on the x-axis here, and we'll have over here magnitude on this axis. Now I'm just going through lymphocytes in general. There are some differences between T and B cells, etc., but we're just talking about in general. So in general, you're going to have a phase that you're going to have activation. And this is usually by an antigen. It can be an infection, it could be a pollen. And then you're going to have clonal selection and expansion. Now, the expansion can't go on forever, and it typically will start to decline at a defined time, regardless of the antigen. So even if you're a chronic infection, acute infection, your immune system works within a defined set of time. Anyways, you have this expansion, and then this expansion will have to go through a contraction. And this is thought to be caused by apoptosis. So we talked about IL-2, so the primary driver of expansion is IL-2. Now the primary driver of contraction is another set of cytokines. We have IL-7 or IL-15. And so these are considered survival cytokines. Whereas IL-2 was a growth cytokine. And then this will eventually plateau and you will have your memory. Now, what's going on in the survival cytokines is once IL-2 is gone, you take away that growth. Now, all those cells that grew have to compete for survival. And there's only a limited amount of IL-7 and IL-15 in your body. And so most of the cells, unfortunately, don't see that survival signal, and they're going to undergo apoptosis. And so that's another way to control T cell activation is you have a mechanism to control the cytokines that are telling the cells to divide, and then you have a competition for the cytokines that are telling the cells to survive. Okay, and that's all. Thank you.